and we hit it up immediately and stayed in touch for forever since then. Like, you know, forever last 15 years. But the point is, why I like to work with John and talk with John a lot is that John has this very broad sort of system perspective on the brain, but then combined with a phonetic dedication to the facts, which drives me mad all the time because too much detail, you know, you get lost. So to find that balance between, you know, a wish and having the concepts right and the construct and to see the system, but in the meantime, we just stay on top of all the empirical facts that we have available. That's quite a balancing act, and John is a master in just doing that. So John, it's great that you're here. John is going to talk about Theta gamma cycles, which is actually a story he's pushing now for a while. The first time I saw you talking about that was in 95 or 96 at the Salk Institute, when you just had your science paper on that. This whole idea that...
Wrong way to 1985.
No. Okay. You see, facts get sometimes in the way of the truth. Anyway, that's another discussion. But the point is that John has been really, you know, driving this whole idea of oscillatory behavior of neurons being key in understanding their function, and it also brought him in conflict with many other people in the field. But John keeps on insisting this is the way to think about the brain. So let's see if he can convince us today. John, great that you're here. Thank you. I'm looking forward to it. Is that me?
Yeah.
Okay.
It's great to be here, and you know, my style is sort of to push a little bit. So I don't like to stick to the facts, but I like to be constrained by the facts and then see how far I can push. And so the direction I'm pushing today is not only is this theta gamma code, you know, relevant for the things that I originally thought, which had to do with working memory, as I'll show you in a minute, but that actually is just a fundamental language of the brain, and maybe no part of the brain can ignore this, and having a sort of universal language, if you will, and then, oh, sorry. Let's see, since I like to do that, maybe I should wear the
mic. Here, I have that. I have it here. Just keep on talking. There's a microphone there as well. No problem.
In any case, you know, Paul and I have been really interested in large-scale integration in the brain, and it would really make sense that there were certain uniform conventions about how to communicate that governed the communication between different parts of the brain. So in the end of my talk, I will have covered two forms of memory. I will have covered a nice example of a sensory system, and then I will have covered a motor system. And my point will be that emerging evidence suggests that something like the theta gamma code is utilized by all these systems. So that's my overall point.
Let me fondle you a little bit, John.
He doesn't run as much as he used to.
I know. Getting soft, you know.
So, maybe that's too loud. Is that all right? I'll talk softer. Is everybody hearing me? Yeah? So my starting point was, you know, thinking about working memory. And so let's do a working memory experiment. This is the famous Sternberg test, which we'll do on you. 4, 8, 7, 6. Was 3 on the list? No. Okay, good. All right, so you held those 4 numbers somehow, then you could interrogate them. And that's what a very famous memory experiment. I'll show you some of the interesting regularities about that experiment, about that task that has been discovered. But in any case, how do you store those 4 numbers? And this is sort of a joke slide saying, well, maybe you have different registers in your brain and you put
the different numbers that I give you into different brain networks. And the theta gamma code sort of struck me as an alternative solution where you use the same network to store multiple numbers or multiple items, but you multiplex. So you use different phases of an oscillation to store the different numbers. So that's a way you can get the same network to be much more powerful. So how did this all come about? Paul is right. I'm sorry. 1995. I thought it was 1985. Okay. In any case, I was sort of staring at data, and the data that you see there is the either intracellular recordings from the hippocampal neurons or extracellular on the top. And so the question is, what do you see in that picture that's interesting? So stare at it.
The world will perish in
No, you
2,000
can't talk.
years. Oh.
You're too old.
I'm sorry.
Everybody else has to figure out. What do you see? Somebody describe what you see.
High gamma band activity during the peaks of the theta band at the top.
Okay, well, first of all, you're seeing two oscillations somehow. And here's an interesting question. Are they interacting oscillations? Or are they independent linear summations? What would you say? How many people are in favor of summating linear? How many people are in favor of interacting? How many people don't have a clue? Okay, you don't have to know any literature.
It looks like they're related somehow.
And why does it look like they're related?
Because you have little dotted lines. Relating them.
Okay, this one is very smart. Anybody? Let's look at the intro, because there it's the most obvious. Is it just a continuous high frequency thing, and then you add to it a low frequency thing? Is that what you see?
I don't think people see what you see, Joe.
Well, okay, it takes a while. You just can't instantaneously perceive the truth. You have to mull it.
No,
They're not interacting.
I guess not. So what you're saying is the first one you have lots of high frequency, the top one, and the lower one is a
No,
low.
okay, so we're just now looking at the lower one.
Just the lower
So there's a low, let's say there was a low frequency thing and an independent high frequency thing. Would that account for this data? No, because what you see is that the amplitude of the high frequency thing is being controlled by the low frequency thing. So actually, it's even hard to see the low frequency thing by itself in the bottom trace. In the upper trace, if you sort of smooth out the high frequency, you would actually see the low frequency. But in the bottom trace, you don't see it that well. But what you do see is that the amplitude of the high frequency thing is varying as a function of time, and that looks like a low frequency variation.
So then, would you say that in the top trace they are not interacting, and in the lower trace they are obviously interacting?
I wouldn't say that, because we'd have to be a little bit more quantitative. It's harder to see the amplitude of the high frequency in the top trace. I think they're always interacting. And then so that's kind of interesting, that it's not just a question of there being two sources somewhere in the brain of these oscillations, but they're part of a unified system. That would be my guess. So now here comes the big miracle, and that's on the right, that I had a crazy idea and that it got published in Science. Who's ever heard of such a thing? It's not even generally possible to get good ideas published in science. Okay, so the idea is that why would you have oscillations in the first place? And so that you could use the gamma cycle, cycle A, does everybody see cycle A? And now you would say, let the network be dedicated to representing the first memory that I'm going to give you, which is, let's say, B. Let me use numbers. What you're trying to remember is 4896. So we're going to, oh, thanks. So we're going to put the number 4 in the network, and how are we going to represent the number 4? So the most classic idea in neural network theory is that you represent something by the spatial pattern of active cells in the network. So, and here you see 4 of the cells that are active in black, and so that would encode the number 4. Now, you're keeping in mind not just the number 4, you're keeping in mind the number 8. So instead of having a separate register in your brain, you use the same network, but now you use the second gamma cycle B, to represent the number 8, and it's represented by the fact that a different subset of cells in the same network are active. And so how many numbers how big a list can you remember if this is true?
7, I was.
Okay. So, I mean, I sort of started with this idea, and then I was lucky enough to have a psychologist, Mike Kahana, down the hall, and he told me about some absolutely classic experiments, behavioral experiments, in the science of short-term memory. And so this is an experiment that has to do with span. And so I don't even know who ever did this experiment. I don't even know if anybody knows who did this experiment. It was done so long ago. I actually think it was done by shepherds who were sitting there taking care of sheep and were really bored, and they said to each other, how many numbers can you remember? And they played this game.
Is that what shepherds do?
Well, that's the good part.
Okay.
Okay. And so the amazing thing is that if I give you a 6 item list of digits, or you'll do pretty well. We could try it, and the likelihood of your spitting back to me a phone number is not bad. And if it's just a little bit shorter than a phone number, you'll all do very well. Like 4896 is just so easy, right? But when I get up to 8, suddenly you'll get really lousy, and that's what this says. The drop-off is really lousy. And so, I don't know, maybe this has to do with the fact that your short-term memory uses a code in which you have a limited number of gamma cycles in which to put different information. Okay, that sort of seems like on the right track, but here's another experiment that kind of suggested to me I was on the right track. And this is the Sternberg experiment. So this, again, you could do if you were a shepherd. So we already did one. 4896 is 8 on the list. Answer as quick as you can.
Yes.
Good. Okay, so I can measure, if you're really motivated, I can measure your reaction time, right? Now, what do you think is going to happen if I give you a 5-item list? Is it going to take you longer to answer?
You're going to try it.
Okay. I can't measure. Anyway, so Sternberg measured this, and he found this beautiful relationship. The longer the list, the longer it takes you, and for each additional item, it takes you, in this case, 38 milliseconds more to answer. So, ding, ding, whose bell is going off? 38 milliseconds. What is that roughly equal to? A gamma cycle, period. So, in other words, Going back to this scheme, the time between this fast oscillation, well, we don't really know exactly what it is. It seems to vary, but roughly speaking, it's 30 or 40 milliseconds between here and here. So you could imagine that there might be some relationship between gamma and this thing. And I think the fact that this idea kind of made sense, and that it made contact with two very fundamental observations in psychology is the only reason I can understand why science took this paper. In any case, what I would now want to do is to sort of bring you up to date. And so we would call this a theta gamma code. And another thing about this is that probably in terms of working memory, since you want to keep all the numbers in mind, 4, 8, 9, 6, if each gamma cycle represents one of those numbers, then you just repeat the whole 4, 8, 9, 6 in the next theta cycle. So this is a way of keeping a whole short list in mind, so that when I finally ask you, was a number on the list, you can interrogate your short-term memory encoded in this way. So, now, when we actually proposed this idea that there might be an oscillatory basis for working memory, it was a very radical idea, because there was just absolutely no indication at the time that oscillations were involved in working memory. Luckily enough, my colleague in the psychology department, Mike Kahana, was at a meeting, a totally chance meeting, where he met Joe Madsen, a neurosurgeon at Children's Hospital. And Joe was saying, I'm implanting all these electrodes in epileptic patients, and they're sitting in the hospital waiting to have a seizure, because then we want to find out with the electrodes where the seizure was so we can resection that part of the brain. And the sad thing is that here we have electrodes in the brain, and nobody ever studies what's happening in the brain in these patients. Would you like to take a look? And now intracranial recording of epileptic patients is incredibly common, but this was actually only the second lab that had ever done this to take advantage of this kind of capability. And so Michael and I, set out to do something that would be interesting. So we said, well, since we have an oscillatory model of the Sternberg task, let's go in and do the Sternberg task on these patients and see what the brain is doing. Are there going to be oscillations? And so, all right, so here's the task. This is a very short task. Only 2 items were presented, and then the test here. So are there oscillations in the human brain related to working memory? You guys don't like to look at data.
It's just a distraction.
There is, because the amplitude increased when the object was presented, but then decreased when it was recounted back.
All right, well, now you're looking at details. So here we are before and here we are afterwards. So what would be the first approximation statement? Okay, guys, come on. Well, let's do it here. So here we have the recordings from 1, 2, 3, 4, 5 trials. Are you excited or not? I mean, it's just it hits you over the head, right? Will somebody please get hit over the head for me? What's the conclusion?
Activity increases during the task and is maintained, and then when the task is done, it goes low.
Okay, here's,
That's what's hitting me over the head.
thank you. Okay, fine, great. I mean, when you consider the fact that people were saying oscillations had nothing to do with working memory, this is incredible, right? This is jump up and down time, right? I mean, and this is not subtle, right? We didn't do any filtration to see this. This is mammoth, right?
But we're not sure yet whether the deflections you see here are really working memory related. It might also just be, let's say, a non-specific arousal effect because you show me something.
Okay, so in fact, Paul oddly is right.
Let this be noted.
In the record book, and correlation is not a very good, but anyway, we jumped up and down. We chose not to be as cautious as Paul, and we said, oh, wow, look at this. This shows, that at least part of what we imagine must be true, right? We see these theta frequency, this is sort of theta frequency, we see these theta frequency oscillations, they turn on exactly at the point where you need working memory, they turn off exactly where you don't need working memory. And so all you naysayers that say, oh, there's no oscillatory basis for working memory, go jump in a lake, right? That's what you would say, right? And that's what we wanted to say. But we were honest enough to say that sometimes we see this. Look at that bottom trace. So what would you say there? So just see, this is the frequency of the oscillation. This is time, and redness is the power. So what would be your conclusion from the bottom trace?
What does the white bar represent? Is that the presentation?
The task.
Yeah, this is the task. Yeah, the task is from here. Oh yeah, this is the task. So this is what I showed you before, but we saw some sites that look like this.
Well, it's the other way around.
Other way around. So which one is involved in the task? Which one do you think is important? Both? Okay. Anyway, we have no idea, right? All we have is cute, basically. All right, so let's fast forward to 2015 and a second line of work with these authors. In particular, important was Josh Jacobs, because he and Mike Kahana had a lot of data, and they also did the Sternberg task. Now, in the Sternberg task, they used letters. So A, U, X, Y. Was Z on the list? Thank you. Now, they said they were interested in a different thing. They said, are there sites that respond differentially depending upon which letter I show? And they published a paper saying, yes, we can find sites that respond differently whether it's an A or an X. And we got very excited by that because, as I said, we really had no idea where in the brain to look for short-term memory. And so, depending on where we looked, we either saw the theta increase or we saw the theta decrease. But we said, well, wouldn't it be a lot stronger if we could examine specifically the sites that had letter-specific function, because those certainly must be involved in the task. Or more likely, Paul will object, but more likely involved in the task. So we got this huge data set, and Ali did the analysis. And the most amazing thing to start off with is, if you have like 40-some electrodes like this, or whatever it is, and you look for these letter-specific sites, you find about 1 per patient. These are incredibly rare sites. So whatever this is, and it tends to be in the fusiform gyrus, these are hard sites to find, but they're really interesting sites. So now the question is theta going to go up or down? And will it have the property that we were seeing somebody articulated quite nicely in those other sites, where it went up and stayed up during the entire period of working memory? Maybe it won't, right? Maybe it'll have some other function.
In the previous slide with the time frequency histogram, were those 2 trials for one site, or were those 2 different sites where you had an increase and a decrease?
I'm sorry.
You had,
Oh, those were different sites.
those are different.
Yeah, I'm sorry, I didn't make that clear. And this is low frequency. This is the theta, not the gamma. And so these sites that Kahana and Jacobs had found that had letter-specific oscillations, the oscillations that were letter-specific were gamma, not theta. So anyway, still very interesting. Let's look at these sites. Now I'm going to show you what the data looks like at these sites. Okay, so first letter presented, second letter presented, maintenance period, test. Okay, you guys are really good scientists now. What do you see? The data is right there. It's just a question of whether you can get your brain to process it. So let's ask the first question. Does this data in any way whatsoever resemble either of those 2 sites I showed you before, where theta either went up or down during the whole test?
Not at all.
Thank you. Paul almost will be an experimentalist soon, or at least he'll be able to interpret data.
I'm doing so well today.
Yeah, you're doing well today, Paul. Does Paul get a grade in this class like you guys? So does everybody see that? That how misled we were by those other sites, which we kind of picked out because we thought they were pretty? So in those other sites, we picked them out actually because the theta or alpha or whatever you want to call it, 8 to 12 hertz, because it seemed to be related to working memory, right? It went on and it stayed on during the entire trial, and then it went off at the end of the trial. Does theta do that here? Can somebody summarize what you see in the low frequency band during this experiment? Not Paul? Write the abstract for this paper. So at these sites we found that low frequency oscillations did what? They decreased, blue is decreased during item presentation, and during maintenance they increased, and during probe they decreased again. So those other sites that we had picked out at random because we thought they were pretty had a very, very different pattern, right? So if you were to look at this at first blush, what would you say about the theta-gamma hypothesis of multi-item memory? Would you be excited or despondent?
Or worse.
Or worse, suicidal or happy? Suicidal or happy? What would you say?
Looks like they're distinct.
Hmm?
Looks like they're distinct.
Well, things are certainly distinct during different phases, but what's even a bigger problem? During maintenance, What do you think about theta gamma coding during maintenance?
There is no theta. There is.
What?
There's no theta.
There's no theta, right?
There's no gamma.
I mean, there's no gamma. Sorry.
Now look, if I would have written a paper published in Science on theta gamma coding just as a thought experiment, my career would be finished, right?
Right.
This is the end.
This is the end.
Yeah.
Paul is going to not make me commit suicide. He's just going to kill me right on the spot. And that would be a service to science, is what he's saying. Okay. But he was wrong. Okay. You know, he can't always be right. So now I'm going to show you that don't be fooled by numbers. You better do a little bit more homework. Okay, so, I mean, we have to look with a specific test to see where is there theta gamma coding. And all we have here is amplitudes, and, you know, they're very surprising. But anyway, we haven't really finished diagnosing this slide. So, does this slide support the original idea of Jacobs that there were gamma-specific, letter-specific gamma sites? Yes, because to this letter you get small gamma, and to this letter you get big gamma. Okay, so are these sites really part of a unified system? Is there anything you could say about the data that would point to the fact that these sites, although this one says, oh, I like this letter, and the other one says, I like this letter. Is there something that unifies these sites, that makes them part of the same system, that is hidden in this data? So the letter system, we'll call this the letter system, has subparts which like different letters, but is still a system because blank, blank, blank. Stare at the data.
What's in the right side?
No, don't, no. We won't do that yet. What do you see here? What shows that this is a unified system? No. Okay. They're not in antiphase, actually. Take a closer look. Well, I guess maybe you could argue. What I'm seeing here is that the low frequency suppression is letter independent. So the whole system says, okay, we're going to engage you, and we're going to shut down your low frequency power, and then the part of the system that's engaged in the particular letter will get gamma, and the part of the system that is not engaged in that letter won't get gamma. But the amazing thing is that in terms of the low frequency suppression, it's the same in both regions, which is an indication that there's a rule that governs this letter system. Now you say, well, okay, let's look at this. This is a site that does not have letter-specific gamma. It has none of this interesting stuff. And just to show you how incredibly interesting these sites are, let me show you every electrode in this patient. Now, I'm sure instantly you'll analyze this, right? So the site that you've seen is up here. So what do you see? What do you see in the data?
The theta is not switched off systematically.
Yeah, I mean, this whole pattern, gamma up due to the right letter, theta alpha down, is something about this system and a very rare phenomenon in all the other sites. So we've honed in on something that is pretty unique to this system, and it's an amazingly salt and pepper system. So sometimes we find more than one site that does this in the same patient, and they're not next to each other. And for sure, if you move one electrode away, which is 1 millimeter away, and say, do you find this pattern? The answer is no. So there's a salt and pepper system that does letter recognition. Okay.
But John, the grid you show us, this matrix, this is representing space on the cortex or
No.
depth?
No.
Or some other...
I think it's organized from the most letter-specific, there's, when you do an analytical measure of letter specificity of gamma, you actually have a graded number. You can get slightly letter-specific or totally not letter-specific, and these are arranged according to that. Okay, so now it turns out that there are analytical methods that look at cross-frequency coupling. Okay, and I can easily explain it to you, but there turn out to be several methods. And it's interesting to see that we had to use both methods in different situations to make sense of this. Okay, so this is a slide that's during encoding. When you give the letter, remember that encoding means that the gamma is going to go up and the low frequency is going to go down. So it's hard to make measurements on the low, right? Because it's gotten so small. So what you do is we do what your eye did in that very first data slide I showed you. Let's just look at the high frequency and see if the amplitude of the high frequency goes up and down at a low frequency, with a low frequency rhythm. So all you have to do is take the envelope of the high frequency and now do a Fourier spectrum of that and see. And so that's the method we use. And here what you can see is that for a tuned letter, so if we show a letter that this site likes, we do get cross-frequency coupling during encoding. And it's a little dirty, but this is the envelope of 80 hertz. That's what we think is the high frequency, and it's modulated by something in the 10 or even 5 to 20 hertz range. So that's the low frequency. So there is a modulation of the high frequency. There is cross-frequency coupling. So why was Paul wrong when he said, well, since the low frequency has gone down, you couldn't have cross-frequency coupling? He was wrong because amplitude of field potentials is, to a good first approximation, meaningless. That is to say, we don't really even understand what makes field potentials. So field potentials could be determined by the number of cells that are active. In cortex, there are many, many layers. Maybe during some condition, all the layers involved. During some other condition, just one layer is involved. So just because amplitude changes doesn't tell you anything really about function if you're deriving the data from field potentials, because it's too lousy a signal. It's not really a biophysical signal. It's kind of some quasi-unknown reflection of the circulating current around a whole bunch of cells. And it's sort of useful to say, what is the average rhythmicity of the cells? But one can't take it very seriously because of the complexity involved. And so what this shows is that if we had just assumed that because the low frequency went down, that it was unimportant, we would have been, in fact, misled like Paul was. That even a very small residual oscillation can be functionally important in controlling the high frequency oscillations. So now Paul will still say, but this is during encoding, and the theta gamma idea really requires that there be theta gamma during the maintenance period, right? Because that's really what's holding the cells. was holding the information after I presented. So now we've gone on to do an examination of cross-frequency coupling during the maintenance period. So what is the maintenance period? 4, 8, 9, 6. Maintenance. Was 3 on the list? She says no, because she held the information during the maintenance period. And I won't belabor the point, but just to say that now, under these conditions, we have gamma low and the low frequency high, right? I showed you that. So now you have to use cross-frequency coupling of a different kind. So what you do is you don't really know much about gamma, but you can sort of try to estimate the power in the gamma frequency range as a function of time. But you know a lot about the low frequency because you can see it, because it's huge. And so you say, how does my measure of gamma power vary with the phase of the low frequency? I can do that calculation very nicely. And when you do that form of analysis of cross-frequency coupling, you see that there are 2 gamma ranges where you have coupling to the low frequency. So I won't go through, and then if you look at a non-letter specific site, it's much less dramatic, and the whole thing is much less dramatic if you look at an untuned letter. So now I am ready to jump up and down with happiness, because there is theta gamma coupling during the maintenance period of a working memory task. Therefore, it is at least possible. Therefore, we have not excluded the model that theta gamma organizes working memory. So now you'd say, well, okay, prove it. And at this point, one still can't prove it. So what you'd like to do is to show, well, while I'm holding 4, I'd like to recreate that picture. I think maybe I have it in the next slide, with single unit recording. Oh, sorry. I'd like to actually do single unit recording and get an animal or a human to hold 2 numbers in mind, and during the maintenance period, I'd like to see that the firing occurs at different theta phase, i.e. in different gamma cycles. And then I would really believe this model, and that experiment has not been done. So people have been playing around and trying to do other tests of this model. And so for instance, you'd say, well, why do different people have different spans, memory short-term spans? So, there's probably some people here who can remember 10 numbers fine, and if you're like me, I can't even remember a phone number, so my span is probably about 5. And so you say, well, let's look at theta and gamma and see if people with a large span have many gamma cycles within their theta, and that's why they have a large span, and I have just a few gamma cycles within mine theta, which is why I have a short span. And people have been trying that experiment, and there have been some published papers saying, yes, you can do this and it works, but I don't actually believe these. I think they're lousy papers. And I think the problem is that the system that actually does the working memory is too small a fraction of the brain to find with scalp EEG. And so now that we have a way kind of to hone in on the right sites by finding these letter-specific gamma sites, we may have a crack at sort of doing this.
But now, John, can you say something about this differentiation you see in the low and the high gamma? Because that seems to complexify the story a little bit.
Right. Yeah. So, I mean, I guess you're saying, why do you have 2 different kinds of gamma? We'll come back to that later. I mean, there's not much to be said, but there are a few things, but they're not worth saying.
Okay. But then the other thing is that now your interpretation, you bring it really down to, let's say, a single cell interpretation. Because you say, look, we got to see that a single cell is maintaining this information
right.
following theta gamma. But as you said earlier, the deflections you see in the local field potential can also be just a population effect.
Right.
So it might also be the case that this information is reverberating in a circuit that collectively is responding according to some theta gamma coupling. So do you go for the single cell interpretation or for the population interpretation?
Yeah, I don't kind of break this dichotomy. I mean, I think this is a network. There's a whole network of cells. I mean, there's a network, and some subset of cells are firing here, and some set of cells are firing here. And now, it's possible that you have an overlapping code, so there's nothing wrong with the idea that some of these cells are shared by the number 3 and the number 6. Oh, I guess.
Mm-hmm. Okay.
So, but still, I mean, you've got to be able to demonstrate some sort of phase coding during working memory if you're going to totally believe this. And this is why I'm now going to segue into long-term memory. And I'm going to argue that in long-term memory, the theta gamma code has now been proven beyond any doubt. And the reason is that we can look at it at the single unit level and make some sense of it. So in contrast to working memory, where the single unit data is not yet given to us, in the case of long-term memory, it has been given to us. So this is a circuit diagram of the hippocampus. You have the dentate gyrus, CA3 and CA1. And this is a very famous experiment from David Reddish. And I like it because it kind of illustrates the obvious, but it's nice always to have an experiment that illustrates the obvious. If you came to a choice point, you would use your memory, to say, well, is the exit on the left or is the exit on the right? And this is incredibly valuable, right? I mean, if in a fire you remember that the exit is that way, it might be life or death. So that's a prediction, that if I go left, I'll come to this, and then I'll turn left, and there'll be a door. If I turn right, nothing happens, or there's a vicious animal down there. I mean, we want to remember what happens at choice points because it's very important to make the right decision. And so you want to consult memory. And so for an animal who reaches this decision point on a maze, it has to go left or right. And in this experiment, nowadays, you can really tell what's going on in the hippocampus because you can record from many, many cells. I mean, the latest technology is you can record from several hundred cells at the same time. And I know you know from Dr. Moser's talk about place cells, but what he may not have emphasized is that by virtue of knowing about the cell's place field, we can actually know what the rat is thinking about when it's not, even when it's not at that place, because the cell will be active if the animal is just thinking about that place. So if you're thinking about the door and there's a place cell that is responsive. So what you first do is you let the animal or yourself, let's put an electrode into you. Okay, so you're running around, you came here this morning, and now we have the huge data set. And we find that one of the cells in your hippocampus like that door. That, you come down that ramp and now you're in a door. Okay, so that's the door cell. So here's the question. Fire, you run back there, okay, and you got to figure out whether to turn left or right. And what David claims is that what will happen based on his, am I not hooked up anymore?
It's on the side.
Sorry. Okay, so let's just imagine that here you are, and you're a rat, and this over here is the door. So what he claims is that he has seen the following, so I now need to understand what's being plotted here. You look at the cells that are firing, and let's say at one moment in time, let's just take a very small time, 10 milliseconds, the cells that are encoding for this place are firing. Then I would put a red dot on that place in that graph. But if these cells were firing, then I would put a red dot further along the path. And if Paul is sitting at the exit, right, and I actually was thinking about the exit, then the Paul cell that was the exit cell would fire. Okay, so here's what he sees. And remember, here the animal is just staying right here. But as a function of time, you can see the active cells are now moving along, not moving, the cells that are active have place fields that are further along this path until finally they even cross the corner. So the rat is imagining this whole path that will take it, in this case, to a goal site. So then he waits, he being Johnson and Reddish, the experimenters, wait a few seconds, and lo and behold, now there's a sweep of activity down the other arm of the maze, down this arm. And so this is just exactly the kind of information you're going to want to have available to your action selection system to know whether you should turn left or right. Now, what I haven't told you is that all of this occurs during one theta cycle. It all occurs in roughly 150 milliseconds. And, as I'll now demonstrate to you, it's organized by the theta gamma code. So, this gives you a demonstration of a case where one of these sweeps started here and then it moved out here. And you can see that here the cells are organized according to the center of their place fields and have different colors. And you can see that the cells fire during the theta cycle in the order of their position along the path. So that's what it means. That's the conclusion you can derive from the fact that this looks like an ordered progression. And you can see in the field potentials both the theta and the gamma, but is the gamma really important? So this incredible paper just came out in Science, which proved that, in fact, the firing of cells is organized by gamma, and in fact, the transition from, let's say, one place being represented to another place being represented is not continuous. So during one gamma cycle, all the cells try to represent a given position, and there is no movement. But then, in the next gamma cycle, suddenly it jumps to represent the next position along the path. And so this paper documents that jumping aspect. So how can you see it? Because here they've coded the position of the place fields that the animal is representing during this period. And you can see that that the activity first specifies this position, but then it jumps in the next gamma cycle to represent a different position. And so the fact that this is not continuous red, but is jumping between points, is evidence that the whole process is being discretized by gamma. So you can say, what's the proof that it's really gamma that's discretizing it? And that proof is in this right slide. So what they did is to say, let's
measure as a function of time jumpiness. So it's not jumping, it's not jumping, it's not jumping. Ah, now it jumps. It's not jumping, it's not jumping. Ah, now it jumps to. Oh,
Maybe I should swallow it.
This time will not.
Okay. Okay. So what here you see is the jumpiness is phase locked to the field potential recording of gamma. So the conclusion now is that yes, we've known for a long time that theta phase is an important coding variable in the hippocampus. This goes back to work of John O'Keefe. But now we know that it is in fact a discrete phase code organized by gamma. You have discrete phases because you have an integer number of gamma cycles within a theta cycle. And so that is the definition of a theta gamma code.
But in some sense this goes deeper, right? Because now you also have a fast gamma that inside the slow gamma gives you a continuous bit of space.
Okay, well,
Or not.
we're going to argue about fast gamma later.
Okay. But the red dots would suggest that, right? You agree with that? Here, the red dots in the top panel would suggest that within the slow gamma you have multiple responses.
No.
Oh, no, okay.
No, the red dots, you mean multiple spikes or what?
Yeah, yeah.
Yeah, no, you can have multiple spikes
Mhm.
within a gamma cycle, and this makes the attractor work, as we showed in our paper together. So just how much time do I have?
Well, how much time do you need?
Okay, I'll
No,
take
look, we're doing fine, John.
Okay, we're doing
We have
fine. Okay.
At least, let's say half an hour or so.
Okay, so this is some work that
Am I wrong?
Cesar
Okay.
and I have been involved in. And I just want to mention it because you can say, well, it's so interesting that we have the ability to look ahead down this hallway, right? I mean, what could be more valuable? And we'd really like to know how we do that. You know, how does the hippocampus do that? And this is just a short aside to say that you heard about grid cells from Dr. Moser, and in the paper that Cesar and I are resubmitting shortly, we claim that the whole function of grid cells is to make possible this mind travel down the hall. Okay, so how does that work? So how do grid cells work? So just to summarize this slide, during part of the theta cycle, the action of the grid cells moves the representation of place from x to x plus 1 to x plus 2 down the hall. I'm now going to explain to you how this might work on a neural network basis. So you have to understand grid cells and the mechanism of grid cells. And everybody was excited about grid cells because they seemed to have the quality of an integrator. Okay, so now I have to explain to you what that means. So first of all, what is the current computational model of a grid cell? And the idea is very simple, that you have these bumps of activity, but you have multiple bumps that are evenly spaced. So why do you have the bumps? So the current theory is that all the cells are getting excitation, but cells inhibit their neighbors with a certain space constant. And a solution, it turns out a steady state solution to this is that you will have a grid-like representation. You will have multiple bumps of activity in the network, and they will be arranged in this hexagonal way. So why is that? Because the inhibition, so here you will have, in this region, cells getting inhibition from both here and here, whereas these cells are far away from other cells that are giving inhibition, so it makes sense that there would be more excitation here than here. So, can anybody, that's not the greatest explanation, but does that give anybody any intuition of why this would happen? Sort of? Okay. Now you could say this is a form of, this is a kind of funny form of reality. Like this thing is just sitting there, these bumps are just sitting there. So now comes point number 2, that one can imagine the special kinds of mover cells which will cause this whole thing to slide. And so this picture is supposed to represent the idea that we're just sliding these bumps along. Now, supposing we make another rule, that the rate of sliding the bumps along is proportional to the velocity of the animal. Okay? Then what we get is that the position of the bumps could represent the position of the animal. Because when the animal moves at a certain velocity, the bumps move in a way that corresponds to the coordinates of physical space. So it becomes like an analog representation of physical space. So now one more step. If these things represent where you are and can integrate a velocity, integrate just means that, if I say I moved, the bumps just keep on moving. So they are integrating velocity over time. Suppose you're standing still and I put a velocity vector into this system. What's going to happen? It's going to make you think that you're ahead of where you are. In other words, the bumps are going to move, and you're going to say, well, I'm not here anymore, I'm here. And then more velocity comes. Well, I'm not here, I'm here. Right? So what we're saying, what Cez and I are saying is, look, this is exactly the kind of system you want to look down the hall. So you're at the hall, you put a velocity into your grid cell system, the bumps start sliding along in space, all of a sudden you're there, now you're finally at the pole door, and you say, oh, there's the door, it's on the left, so your decision system says, I'm going to go left. Anyway, so I just wanted to connect this idea up, saying that we have, I'm saying that during theta gamma, you code for the path down to the door, and that we actually have some ideas now of how the entorhinal grid cells might be responsible for that process. That's what I wanted to get across from you. So that was a little diversion there. So now we're going to come back to just say that, as Paul says, I don't like to totally bullshit. So we're coming back to an experiment. So, what would Cez and I predict if you got rid of the medial entorhinal cortex where the grid cells are? We'd predict that you could no longer mind travel down the hall, and the Leutgeb paper used something called phase precession, which is a little bit different, but it's also an indication of this traveling process. I won't go into the details, and concluded that this traveling process was no longer evident after the lesion of the medial entorhinal cortex where the grid cells live. So, we think we're on the right track in understanding, what the grid cells might do to implement this important function that is mediated by the theta gamma code and allows you to very quickly look down one or other of the halls.
But now is it completely disappearing, or does it get more, let's say, more noisy?
It
Right.
gets noisier,
It
but you
seems
know,
to be
you
getting
don't see,
more-
you don't, it's the slope which is gone.
Mhm.
Anyway, there may be a little residue, but not much.
Okay.
So now I'm going to switch gears and say, well, okay, I've emphasized the theta gamma code in the context of two types of memory. Short-term memory, interesting stuff, no definitive data for the theta gamma code. Long-term memory, definitive data. Okay, now we're going to switch to the question, could it be that theta gamma code also is important in sensory systems? And the answer is going to be yes, but I'm going to restrict myself to a discussion of recent work in the olfactory system. So the olfactory bulb is the structure in the periphery, and it sends axons back to olfactory cortex. And it's been very difficult to study the olfactory system really in a behaving animal, but that problem is now solved. And so people like Dmitry Rinberg, whose data I'm showing, have now made it possible to see what's happening in the olfactory bulb output from in an awake behaving animal that's sniffing. So you automatically say, well, of course it's going to be oscillatory because the rat sniffs at theta. So, okay, how could it not be an oscillatory process? And I'm just going to show you a little bit about what actually happens and why it kind of makes sense. So what you see is that if you study a given cell to a given odor, and this is now a very high time resolution picture, and look at the spikes coming out, that during this sniff cycle, which starts here and ends here, that this particular cell gave a burst with a very time-locked phase during the theta cycle, during the sniff cycle.
So this is mouse or rat?
This is rat.
Okay, and then we're in olfactory bulb or mitral cells? Or?
This is mitral cells.
Okay, so they're reading out the glomeruli.
Right.
Okay.
So then the next question is, do different mitral cells have different preferred sniff phase? So here you see the distribution of sniff phase, and you find, oh my God, different cells will put out these bursts at different part of the sniff cycle. So each one of you might be tuned to fire at a different part of the sniff cycle. Now, this is now what's the next question? Let's change the odor. And if we change the odor, then it is still true that each of you is going to be locked to a given part of the sniff cycle when you give out a burst, but it's going to be completely different if we change the odor. So this is incredibly rich information to see the pattern in which all of you fire, and we're going to change from a Bordeaux wine to a Merlot wine, and this richness of all you guys firing at slightly different phases is going to wind up being able to tell the central nervous system whether it's a Merlot or Bordeaux wine, right? And that's what a really good wine taster can do. So that's how we think the system works, that it's the sequence coming out that defines the ultimate odor during a theta cycle. Does this have anything to do with a theta gamma code? Well, you can answer that question by finding out if the bursts are time locked to gamma. So with Honey, Sanders, and Kulikov, we got Rinberg's data. We got the field potential, so we knew when the gamma cycles were, and now we know when the bursts are, and we could ask, do bursts occur time-locked to gamma phase? And the answer is yes, they do. So this is a theta gamma code. Different information is in different gamma cycles. But, to know whether it's the Bordeaux or the Merlot, you have to decode the sequence to know which wine it is. Still, so this is exciting. But in the next minute or two, I want to describe a theoretical paper that we did, which said, well, if the fundamental problem of recognition is to recognize a sequence, which is carried by the theta gamma code, is there a simple way to decode it? And we built a network which does it, and it's not that hard. So what is the classic way that you guys have learned to recognize something? Maybe you learned it in your neuroscience class. Well, you learned about Hopfield nets, attractor networks, and those say that if you have a certain spatial pattern, then you can recognize that pattern as Merlot, a spatial pattern, whereas a different spatial pattern of cells would encode the Bordeaux, right? And then these can form attractors, and if you don't know anything about this, ask Paul. Okay, so you could state the problem of sequence learning as follows. You could say, if you could convert a temporal sequence into a spatial sequence, then we could use the classic ideas of recognition based on attractors to know whether it was a Merlot or a Bordeaux, right? So how can we convert a temporal pattern into a spatial pattern? So in the solution that we've come up with, we solve it in what we like to call brute force methods. Nothing sophisticated here, just complete brute force. So what are we going to do? We're going to divide a network into modules. And every cell gets input from the olfactory bulb. But these cells start out being capable of bistable structure. So if you get them excited, bistable means if you get them excited, they'll stay excited. They're like a switch. These cells here in the second and third module are not bistable. So when the input comes, they may fire a little bit, but they won't fire persistently. They don't have this working memory switch-like function. You with me so far? But what we say is that these cells, when they're active, can prime these cells so that now they become bistable. They become capable of working memory. And therefore, when the second gamma cycle comes, these cells, which got input during the first cycle but couldn't remember it because they weren't bistable, now they're bistable, so the cells that get input will not only fire, but persistently fire. And they will prime this guy. And so, in the end, what you have is a network that looks like this. So, you have a group of cells in the first network, the first gamma cycle excites them and they stay active. Then they prime the second one, so now the second gamma cycle comes and they excite the cells in this module and they stay active. So the cells in each module are representing what happened only during one gamma cycle. And so by the end, you have a spatial pattern here which represents the exact properties of the sequence. And so now you can say, well, so let all these cells be an attractor, and that will represent whether the wine was a Merlot or a Bordeaux, that pattern. So if we had put on if we think of this as a Merlot, with a Bordeaux, maybe instead of some of these cells being active, some of these cells might have been active. And so you can tell the difference. But those were the cells that were different in the first gamma cycle. Or maybe it was cells in the last gamma cycle that were different. In fact, I think it's sort of intriguing to me that, when you listen to people describe wine, they describe it as a temporal sequence. It starts out this and it evolves to that. I don't know. Anyway, that may be just a coincidence. So what's my point? My point is that here we have an example of a odor not being represented in the olfactory bulb by a spatial code, but by a theta gamma code. It's the temporal sequence which defines the odor. It's definitely utilizing this theta gamma idea, but using it in the context of a sensory system. And it's not unreasonable because there may be straightforward ways, as I've illustrated here, to decode the sequence. So I'm going to finish my talk by going to the motor system and saying, well, is there any reason to think that the theta gamma code might be important in the motor system? And, well, what happens if you put an electrode into the motor cortex? Well, lo and behold, you see both theta and gamma, so it's got to be fairly reasonable to think about theta in the motor system, but that doesn't prove that the theta has anything to do with actual motor control. And so here's an experiment where you have your hand on a cursor, and you're trying to, on a what?
London, a joystick.
On a joystick, and you're trying to move the cursor around the semicircle without hitting the walls, which the person does not do in this first trial. It actually goes through the walls, but over time, the person gets better and better. Now the question is, do you think that this is a smooth motion? Well, now that you're quantifying the motion electronically through the joystick, it becomes very easy to look at it. And with 3 different measures of the jerkiness of the motion, so it can be quantified by this thing called jerk, or you can just measure the acceleration, it turns out that there are oscillatory peaks at about 10 hertz at theta. And so if we would look microscopically at this, it really is that when you look closely. It's not a continuous process. The motor system is discretizing the commands at 10 hertz. And now we come to the most amazing thing. It's not surprisingly that over time you get better, but the most amazing thing is, does the 10 hertz change as you get better? Or if I force you to do it slower, I say, please do this very carefully, do it slow. Is that going to change the 10 hertz? And the answer is, nothing, absolutely nothing affects that 10 hertz, right? No matter whether you're at the beginning of learning or the end of learning and your behavior is very different, you're locked at that 10 hertz frequency. This is actually the duration in milliseconds. And if I ask you to do it slow or if I ask you to do it fast, doesn't matter. It seems like your motor system is locked once every 120 milliseconds in this case, 140 milliseconds. Once every 140 milliseconds, I'm going to make some sort of correction. So this is a pretty interesting demonstration that those oscillations in cortex might have a function in discretizing motion. But how do you know that the discretization of motor action has anything to do with actual processes in motor cortex? Is there any way you can really nail that? Well, the way you nail it is to record from motor cortex and then find out whether those discretizations that you see are phase locked to the oscillation that you see in motor cortex. And so here's an experiment that shows when you average across trials, here's the onset of the motion in a monkey, or is it a rat? I don't know, this is, do you remember? Is this monkey or rat?
That's the Foucault paper?
Yeah.
No, that's a rat.
That's a rat. And you record the theta. It always starts here at the trough of the theta in this example. the trough of the ongoing theta. So this is, I think, the strongest evidence that the motor cortex is really oscillating at about 10 hertz and sending out motor commands phase locked to that oscillation. So I think I'm going to end here just by trying to give a summary that we sort of started out with this idea of the theta gamma code for working memory, but fundamentally what it really said is, well, if we have to store multiple pieces of information to make something interesting, like what list did you just say, then you want to have a data formatting system that allows for that kind of information storage, and the theta gamma code seems to be up to the job. In the case of long-term memory, we now know that it is not only up to the job, but it actually does it. That we can actually see different cells that are systematically active in a sensible way in different gamma cycles. But, as I sort of suggested at the beginning of this talk, when nature sort of figures things out, like I want to send you a multi-item message, maybe it uses that in other situations. So then I presented some of the work on the olfactory system. So what's the multi-item message there? Well, we don't really know how to describe it. We can say some parts of the olfactory, some mitral cells responded early and wanted to send an early message. Maybe for when we get to understand the system better, we'll understand what those cells are saying. Maybe it's first a statement about, well, it's not acidic, right? Or something like that. And then in the next gamma cycle, you learn more information. Maybe it's, and there's a little bit of sweet. And in the third gamma cycle, you get more and more refined. And finally, in the last gamma cycle, yeah, but there was a little bouquet of ginger or some stupid thing like that, like these wine people talk. And then finally, from that whole, formatted information, from the gamma, theta gamma formatted information, you can take that and figure out that it's a Merlot wine. So now we come to the motor system and say, well, the motor system seems to use this, and here we have the least idea. Why exactly is motor cortex wanting to send a multi-part message to the periphery? Well, it could be as simple as, lift and then lift a little bit more and then turn a little bit, and maybe this is important for a violinist or a guitar player. Exactly how you shape your force is going to determine a lot about the beauty of the music that comes out. I don't know. Something like that may be why you want to send a multi-part message every 140 milliseconds down to the periphery to do the shaping of the motor act. That's just total speculation. But I think you can begin to see why, it's not completely speculative to think that this theta gamma code, which started in one domain, may actually be a more universal domain for the representation of information. So with that, I'll close. Thank you very much.
That's great. So, John, which audience member has the first question, you think?
I'm looking that way.
All right, question
go
for
ahead,
John.
Paul. Go ahead, Paul. Oh.
All right, so we go for Ivan there in the back.
So, one question about the hierarchical nature, or does this kind of code not put a limit onto the hierarchical, the number of levels that you can have? Is like, can you have sequences of sequences? Because how you embed I don't know if you get my question already.
I understand.
Okay.
Well,
You translate it, John?
Yeah.
Well, let me give you a specific example, see if you like this. There's one line of thought that the theta gamma code really just captures part of even a yet larger coding scheme that involves even slower oscillations in the delta frequency range. So, I mean, one nice word is the word nesting. You nest multiple gamma cycles within a theta cycle. And then Charlie, what's his name? Charles, anyway, I can't remember his last name now, has been proposing that you then nest theta cycles within a slower, like 1 Hertz oscillation. And that's possible. And my only objection to that form of nesting is that we're now getting outside of the domain of normal cognition. I mean, I think the wonderful thing about the theta gamma code, it says within 140 milliseconds, I'm going to send you an important packet a multi-item packet of information. Well, 140 milliseconds is a reasonable fraction of a cognitive time. A cognitive time meaning almost any task we give you, you're going to do it less than a second. So, the Sternberg data that I showed you, I mean, that's pretty complicated. Searching memory and finding control your motor system, and you do it in 500 milliseconds. So what are we talking about when we say, oh, we're going to nest theta within delta? It just doesn't begin to make any sense because cognition is too fast. But you may have been talking about, hierarchical networks. And, you can always chunk. And that's probably, what's going on in the brain. So you say, well, a sequence of sounds is what you get from the auditory system, and they may be syllables. And all of a sudden that gets chunked into a word. But then the words keep flying at you, right? And so maybe in your working memory now you start having this higher hierarchical level where you put a word representation in the first gamma slot and a different word in the second, and now all of a sudden the theta cycle is representing a sentence, right? And then you chunk those together and you get everything your mother ever told you, right? The ultimate truth. So anyway, interesting ideas to explore.
So, John, regarding the mental traveling in grid cells, where do you think the virtual speed vector or signal is coming from, and who is determining that?
So we have a pretty interesting idea about that, because we think it might be what are called head direction cells. And our thinking, which now has to be revised a little bit, is that actually it turns out that when people looked very carefully at head direction cells, they actually were head direction cells and not direction of motion cells, which it's kind of funny. They called them head direction cells, but everybody thought they were direction of motion cells. So that they would, if the rat was going this way, these head direction cells would, the ones that were sensitive to this direction would fire. But what happens if you're going this way and you're looking that way and wondering, oh, I got to get out of this room. Do you think I should turn left or turn right? Right? And you turn your head, and it turns out that these head direction cells actually are head direction cells. That is, they point in the direction of your head, not in the direction that you're running. As if you move your head because you're interested in what's going over there, and then you put that velocity into your grid cell system and explore what happens if you go in that direction. Now, the complication is that it turns out that somebody really did a nasty thing to a rat, and that it trained the rat to turn a little handlebar and made the rat go backwards. And so now the rat is very concerned about what's behind it. And if you study the mind travel, then it turns out that, in fact, the rat is trying to figure out what's back there. But we had thought that those particular head directions would now point backwards, even though the head was pointed this way. And they don't. Now, it turns out there are many different head direction cells, and so it's still possible that, in different parts of this system. And so this was only examined in one class of head direction cells. It's possible that there'll be another class which does point backwards, so it's an open question. But we like the idea that head direction cells, which, by the way, don't encode velocity, and so are not appropriate for path integration, would actually be important, reasonable signals for mind travel.
So John, let's look a little bit at the source of theta gamma, right? So in case of gamma, I think the current belief is this is very much a product of local interactions between excitatory and inhibitory cells, in particular fast inhibition mediated by GABA A.
Right.
So also in your mind, across the nervous system where you see these kind of signatures of gamma, you always think about these pure local circuit interactions generating it.
Right.
Okay. But now for theta, what's behind that?
Well, we only know the answer in one case, which is the hippocampus, where we have the medial septum, which oscillates at theta and then sends information to the interneurons in the hippocampus, which then control the pyramidal cells and say, shut up, and they shut up, and then the inhibition goes away, and you get all the gamma stuff, and then you say, shut up, and that's how you get the theta modulation.
It's a bit like our usual conversations, no?
No. You say all the shut up, and I have to just take it.
No, but this is now interesting, right? Because it means that theta seems to be more dependent on sort of global regulator, a global pacemaker.
Right.
Right? So now, would you think, if you look at olfactory or motor, that this would be a common source of this pacemaking? Is this linked in some way? Is also the substrate comparable? Or would these all be independent?
Okay, well, you touched something that's really not known. So there are two things to say. One is there are regions in front of the septum which are more cortical, but which are thought to the nucleus of Maynard, I think it's called, which sends cortical projections which are probably theta-like. But I think that the big mystery is the thalamus, and that this alpha-like thing is coming. Alpha, theta. We don't really know how to differentiate alpha and theta, and we don't know. I showed you those beautiful signals, right? That big blue thing, the theta went down. What's controlling that? I have no idea. But my guess would be that it's something to do with the thalamus. And, beyond that, almost
but
nothing.
that then would not be true for the olfactory bulb, because it completely bypasses the thalamus,
No, it's,
right?
well, of course.
But it's interesting, right? Because you see, the inferior olive might be such a pacemaker for your cerebellum.
Yeah.
Right? You have your septum for your hippocampus,
Right.
thalamus for cortex, and then, let's say, some unknown nucleus which will identify towards your olfactory bulb.
Right. Okay. But remember that, in the case of the olfaction, we know a lot more, because in a sense there, it's a sensory motor system, right? The whole breathing apparatus is involved in this low frequency oscillations. The same thing with whisking, when the rat whisks. These are incredibly rhythmic things right at the start of it. So then you can say, well, what about vision? So I was just in Toulouse on the way here. So their huge interest now is, what is the role of alpha in just normal vision? So is vision rhythmic? So this guy, who's been the leader of that group, has shown that if you set up the conditions fairly right, you can get a wagon wheel illusion with normal vision. So what is a wagon wheel illusion? Well, we've seen it all the time in the movies, right? Where the wheel, because of the discretization, because movies are not continuous, a wheel which is turning this way can suddenly turn this way. And you can set this up with strobe lights if you want. It's because even though it's turning this way, you've turned the system off, And the next time you turn the system on, this one is now active, but it is actually in the wrong direction. So the car is moving this way, but the wheels are turning this way. Does this make any sense to people? So, I mean, if you can generate the wagon wheel illusion in a normal human being, you could say QED. A continuous system will not do that. And as it turns out, this guy Van Rundelen is, in the middle of a debate. He thinks it's QED, but other people say, oh, you have adaptation. Maybe it's a continuous system with adaptation. Maybe the adaptation gives you some sort of funny kind of frequency thing. But then he has all kinds of other experiments that, and Ole Jensen has also done these experiments. So supposing I just measure your alpha in your visual cortex, and now I time a flash at the peak or the trough of the alpha, will you see it differently? So the answer is yes, but it's a small difference. So the proponents say, wow, look at this. Obviously your visual system is oscillatory, and the people who don't like it say, wow, look at this. You only got a 5% effect, so obviously you're full of shit. But then it is true that, when you're measuring EEG from the scalp, you're not really measuring alpha in the visual cortex directly, and you have a lot of noise, so you can argue that your inability to get strong modulation is due to your inability to measure the alpha correctly on which to deliver the stimuli. So, if anybody has access to an epileptic patient with electrodes in visual cortex, I don't know anybody here who might be doing that kind of experiments. It's extremely rare to get a surgeon to put an electrode into the visual cortex. So if you ever hear of a surgeon who's doing this, then this is the experiment we want to do. Because now you'd have a good measurement of visual cortex alpha, and we could really then finally see whether delivering visual stimuli at the peak or the trough would make a big difference. Anybody here work on
Volunteering?
Oh, that's right. If you volunteered, there are crazy people who put who put holes in their head and like to stick things into their head.
You can sell it as a form of body augmentation. I think young people will go for that.
All right. Okay. Yeah. Would you like to have your visual cortex augmented? Everybody, raise your hand.
All right, John. Thank you very much.
Okay.
Great presentation.